Thank you, Dan. Our next speaker will be Kim Margolin, who is going to represent the Melanoma Committee as Chair, Melanoma TM Subcommittee. And then we'll have a panel discussion.
Thank you very much. It's a pleasure to represent the Melanoma Committee, and really we'll go back from this whirlwind that you just heard from previous speakers, especially Dan Hayes, to just talk about a couple of studies. And I'm actually going to talk more about the laboratory rationale for the development of these studies, with a lot less about the correlates themselves than what you've been hearing. So maybe you could relax a little bit. The two studies that I'll talk about are the one that has just opened and one that will be opening soon. Same comment about the government review. And they're really both directed at the problem of BRAF inhibitor-resistant advanced melanoma. So just to remind you that approximately half of cutaneous melanomas have an activating mutation of BRAF, and that confers sensitivity to a variety of drugs that inhibit the MAP kinase molecular pathway. So the first study is under principal investigator Alan Alghazi from UC San Francisco, and that will be a phase 2 randomized trial of intermittent or continuous dosing of what we now call combi therapy, dabrafenib and trametinib, a BRAF inhibitor and a MEK inhibitor in BRAF V600E or K, the most common activating mutations, advanced melanoma. The second trial that I'll talk about is from PI's Anthony Rebus, the head of our committee, and his junior colleague Bartosz Chmielowski, which is a phase 1-2 trial of an AKT inhibitor, a pan-AKT inhibitor, plus dabrafenib in BRAF V600 mutant cancer, meaning first the phase 1 will be cancers in general, and the phase 2 extension will be melanomas, and they may have become resistant to either single MAP kinase inhibitor or, again, this combi therapy that we're seeing used more often. The murine model for vemurafenib-dependent drug resistance that underlies the rationale for Alan's study is from data that were recently published and got a lot of press from laboratory studies of a patient-derived xenograft line, HMEX 1906, that showed great deal of sensitivity to vemurafenib at clinically achievable levels. And what we generally use as a readout in these cells is ERK phosphorylation, which is downstream in the MAP kinase pathway. And as you can see here, there's an interesting phenomenon. This is the dose range, and those cells that were parental and were still sensitive to vemurafenib treatment of the mice bearing these cells showed more or less of a linear dose-response curve in relationship to the concentration of vemurafenib, whereas the drug-resistant cells that outgrew in some of these mice actually showed an unusual pattern in which at intermediate exposure concentrations of vemurafenib, there was a sort of a dose-dependent increase in the growth of the cells, and then at higher concentrations, which are not clinically feasible, there was then a reversal of that resistance. So the experiment that was done in a paper that was reported in Nature Letters very recently was to take melanoma from a mouse that had outgrown very early on a resistant or a drug-dependent metastasis that were growing, and then subclone that tumor into many different mice and then studied the clones. And what they showed was, and this was a group from San Francisco and other places, was that there was a major survival impact of intermittently delivering the vemurafenib versus delivering it in this continuous fashion that had produced this drug-dependent resistance pattern. And these two are just the two concentrations using steady dosing that show that the survival of these animals decreased over time. You could overcome some of that with a higher dose, and then the intermittent dosing seems to be irrespective of the dose and produces a much better survival curve. So similar data were actually seen in a group that looked at 42 actual human patients receiving vemurafenib therapy for their metastatic melanoma, and showed that when those patients developed resistance and came off the drug, as shown by this pattern of growth of tumor cells, there were certain ones that had a pattern in which the rate of tumor growth went down, and that was actually 75% of the patients. Only a quarter of the patients showed an immediate growth. So this is very different than what we know about some targeted drugs, and it almost calls to mind the pattern of anti-androgen therapy in prostate cancer, which we learned about a number of years ago. And I don't think this is something you should do at home, but it's certainly worth studying. The mechanisms of resistance were probed quite thoroughly in this study, and as expected, there were no new mutations of BRAF. So these BRAF inhibitor-resistant behaviors in a number of human studies have never shown a gatekeeper mutation such as we see with other kinases. There were no new mutations of a number of other genes that are known to mediate resistance in patients becoming resistant to these therapies. And what they did find was an increased BRAF gene copy number and correspondingly mRNA and protein expression. The characteristics of the cell lines were altered morphology upon removal from vemurafenib and a dependence on not only the presence of the continuing vemurafenib exposure, but also they demonstrated that this dependence of this phenomenon required the presence and the activity of the V600E activated BRAF. So this was based on RNA silencing experiments showing that full knockdown suppressed all proliferation and partial silencing reversed the resistance to vemurafenib. And there was a fitness deficit to these cells in the absence of the drug. So really a very elegant set of studies that underlie the rationale for this study. In addition, Roger Lowe, who is a dermatology partner, molecular dermatologist partner of Anthony Ribas at UCLA, there's also some preliminary data suggesting that mechanisms of resistance that we've seen in humans, in those who develop resistance to vemurafenib, BRAF inhibition, are also in play when patients develop resistance to the MEK inhibitors. So this all looks like MAP kinase pathway-based resistance, and the possibility of overcoming that with intermittent dosing would be of great interest. The highlights of the study then are a 10-week lead-in with continuous dosing, based on the fact that we know that there's a very short time to best response, and hopefully within that early time period, given that the average response interval is about 6 months in these patients, that we shouldn't be seeing an early emergence of the kind of late resistance that was not demonstrated in the study that I just showed you. A 3-week treatment break should allow for the avoidance of, again, the resistant clones and the more traditional resistance, but still the time required to wash out the two drugs adequately. And in this study, the primary objective will be progression-free survival, where the continuous dosing arm, of course, is the control. And it's a randomized phase 2, so each arm will technically be analyzed separately to look for a difference of 13 versus 9 months of progression-free, which would be in accordance with what we know about combined blockade. We'll look at a little bit over 460 patients in this study, and the expectation is that about 3/4 of the patients will have pre-therapy biopsies, and only a quarter realistically will probably undergo paired or on-study biopsies. The idea is to identify differences in the mechanisms of resistance that ultimately always emerge in the two schedules. That will include looking for the amplification, quantitation of mRNA and protein, sequencing for additional oncogenic mutations, and IHC for these intermediates in the pathways. Briefly through the second study, S1221, which is a study that's actually already opened. It's a phase 1/2 study in which the phase 1 aspect of the study will be for any BRAF V600 activating mutant cancer, regardless of prior therapy or histologic type. We think that the vast majority will still be melanoma patients. And the phase 1 will alternate escalations of each of these drugs, the pan-AKT inhibitor and dabrafenib. The phase 2 will then be limited to melanoma patients at the recommended phase 2 dose, as long as the minimum dabrafenib dose in that cohort is the standard dose or higher. The toxicity so far that we know from the AKT inhibitor is predominantly mucosal, GI, diarrhea, nausea and vomiting, and hopefully that won't overlap too much with the dabrafenib toxicities, and hopefully then the mechanism of resistance will be complementary to each other, or the mechanisms of protection against resistance. Very quickly, this is a very busy slide, but I just want to point out that this slide has literally been growing by the day, and those of us who go to melanoma meetings have to fit more and more mechanisms of resistance and little diagrams into this slide. We have NRAS mutations, we have amplification of BRAF, we have MEK mutations, and we have alterations of the gene for BRAF that cause it to more efficiently dimerize, that overcomes the sensitivity. And then there was recently shown in a lab a targeted mutational strategy in which you can actually produce gatekeeper mutations, but we're not sure if they actually occur in the real world. So this is just a picture of the percentage of types of mutations that have been shown in all of the studies pooled together. The core pathways are those of the MAP kinase the core anomalies are those of the MAP kinase pathway, where that comprises about 2/3 of the mechanisms. Another quarter is from alterations of PI3 kinase and AKT, and then what is as yet unknown is 10%. That piece of the pie is going to be shrinking over time. And then from the core pathways, if we break those down, we see this is all MAP kinase here. So again, about 1/3 are from the amplification of BRAF that I mentioned, another 1/3 from NRAS mutations, another 20% from this alternative splicing, and then what we didn't talk about but is important is alterations in the cyclin kinase inhibitors, which comprise about 10%, and a few cases of MEK mutations. I'm just going to briefly look at this. This is adaptive resistance to BRAF inhibitors that occur through alterations of this important parallel PI3 kinase pathway that's shown briefly here. And these are some of the functions: gain of functions, loss of function, mutations. We've known for a long time that many different kinds of alterations of PTEN function can give rise to downstream effects in that pathway, but they all lead to oncogenic cell proliferation and survival, and the endpoint in many of these that we look for is phospho-6 kinase changes. Evidence that AKT inhibition delays the emergence of acquired resistance was shown in Antony's group in a BRAF-driven cell line, and these are graphs of the cell's viability in vitro, where this is baseline after dabrafenib, and then at the time of resistance, again the same baseline, dabrafenib plus trametinib. 2 inhibitors of the MAP kinase pathway in this case didn't really make much difference, and the resistance occurred at day 41 exactly the same. If, on the other hand, you add to that combination this AKT inhibitor, you see dramatic increase in the initial cell reduction, and then nothing emerging in the way of resistance all the way out to 99 days. So the laboratory correlates to this phase 1/2 study will be, of course, the traditional pharmacokinetics that we do in phase 1, and then Sanger sequencing for mutations in the same in those same species that I mentioned to you earlier that mediate MAP kinase and PI3 kinase pathway mechanisms of escape. Quantitative PCR for genomic DNA regarding the mutant BRAF amplifications. IHC for these downstream phosphorylated species, qPCR for the truncation mutation that I mentioned, and then additional studies of these receptor tyrosine kinase mechanisms for escape from MAP kinase pathway inhibition, as well as PI3 kinase pathway inhibition that include PDGFR beta, EGFR, c-KIT, c-MET, that'll be done by IHC, qPCR, and phosphorylation of the proteins. And then, of course, if there are fresh tissues available, the whole exome sequencing and RNA-seq that you've been hearing about. So with that, I'll stop. I'm sorry I had to speak very quickly, and this was a picture at the end of Alan's slide set. This is where we'll be next April, and thank you very much, and have a great SWOG.